Welcome friends. In this video, I'd like to discuss lac Operon. The lac Operon has two words. One is the lac, another is the Operon. Lac here stands for lactose, and Operon is the genetic unit of DNA molecule which regulates the metabolism of the lactose. So lac Operon is a genetic unit to operate, to regulate the lactose metabolism when the prokaryotic cells, that is the E. coli, is taking the lactose as a food. When the lactose is present in the medium and when it is taking it as the food, is then it is metabolizes, it gets metabolized into glucose and galactose to release energy, and that is operated by a genetic unit which is known as lac Operon. So lac Operon is the genetic unit. It is the genetic unit which regulates lactose metabolism when the lactose is present in the medium and it is used as the nutrient by the bacteria. Then this genetic unit, that is the lac Operon, regulates lactose metabolism, and it breaks up lactose into glucose and galactose to release energy. To release energy, the lac Operon operates. Operon means, which literally means to operate. So it is regulated and operated by a genetic unit for the lactose metabolism into glucose and galactose to release energy. So lac Operon is a genetic unit of DNA molecule which regulates, which operates the genes to metabolize lactose into glucose and galactose to release energy. It consists of the lac Operon. It is the first Operon which has been discovered by Jacob and Monod, for which they got a Nobel Prize. Jacob and Monod in the year of 1965, they won a Nobel Prize. They won a Nobel Prize for the lac Operon in Escherichia coli, that is in E. coli. In E. coli, this has been discovered by the scientists Jacob and Monod in the year of 1965, and they got a Nobel Prize for that. Now the structure of the lac Operon. The lac Operon consists of a cluster of genes which are differentiated to perform gene expression of the enzymes required for lactose metabolism. So structurally, the lac Operon, structure of lac Operon, it consists of a structural gene. The structural genes are in three groups. They are the Z, denoted as Z, Y and A, synthesizing different types of proteins or the enzyme. That is followed by another group of genes known as operator, and upstream operator is the promoter. Upstream operator is the promoter, and slightly away from the Operon. The operon has three components: promoter, operator, and structural gene. So there are three components, three parts of an operon in lac operon. The second is the operator. This is operator and this is the promoter. And slightly away from the main body of the operon is a regulator gene, and this regulator gene synthesizes a messenger RNA to form a protein known as a repressor. A protein repressor which stops gene expression is a protein which is synthesized by the regulator gene. So regulator gene regulates the operon, and more specifically it blocks the operon when it is on the operator. It stops operator, it blocks gene expression. So repressor is synthesized by a group of genes known as regulator gene. So structurally it consists of a structural genes, operator and promoter. The structural genes are three in number. They encode the different types of messenger RNA. They are in three groups denoted as Z. This part of the structural gene synthesizes an enzyme known as beta-galactosidase. It is a tetramer consisting of 4 units. Is a tetramer protein, and this beta-galactosidase, it breaks up lactose into, it cleaves lactose into glucose and galactose. Whereas, and some amount of beta-galactosidase also converts the lactose into another isomeric form which is known as allolactose. Allolactose is the another isomeric form which is converted by the beta-galactosidase. Now the Y gene synthesizes a protein, and that protein is known as permease. It is a membrane protein, and this permease, it permits the entry of lactose into the cytoplasm. Entry of lactose into the cytoplasm. And the third is a gene, and that synthesizes a protein known as trans-acetylase, which transfers acetyl group from acetyl coenzyme A into transfer of acetyl group from acetyl coenzyme A to beta-galactosidase. It is also known as thioglycosidase. So the beta-galactosidase is a tetramer, the permease is a monomer, and the transacylase is a dimer consisting of 2 units, 1 unit and 4 unit is the beta-galactosidase. So these enzymes are produced by, this is messenger RNA, and these messenger RNAs are producing beta galactosidase. This is beta galactosidase permease and transacylase. So the structural genes, they produce 3 types of enzymes for the degradation of the lactose and for the entry of the lactose into the cytoplasm of the cell, and transacylase is not directly connected with the lactose metabolism, but somehow it also helps in the entry of the lactose into the cytoplasm along with the permease. Now the operator, second part is the operator. Operator is the part of the operon which switches off or on gene expression, and the promoter is the part of the operon which has 3 sites. For the 3 sites are the recognition site, binding site and the initiation site. These sites are for the RNA polymerase to start gene expression. And I have already told that the regulator genes produces a repressor. And in lac operon, this unit consists of 503,000 base pairs. So the lac operon consisting of a structural gene, an operator, a promoter and a regulator gene. Now the regulation of lac operon. Lac operon is functional. It metabolizes lactose only when the lactose is present in the medium and no glucose is there. When the lactose is present in the medium, then only the lactose, the lac operon functions. It shows the regulation of the lac operon can be explained as follows. Number 1, the lac operon is a negative inducible. It is negative inducible catalytic operon. It is negative means it uses. Negative means the repressor is there to shut off, to block gene expression. It is negative, and the repressive molecule is a repressor. I have a video of operon concept in that the negative and the positive regulation has been explained in detail. Inducible means to start it requires an inducer, and catalytic means it breaks down lactose into glucose and galactose to release energy. So lac operon is a negative inducible catalytic operon. Now when it shows two conditions. When the lactose is not in the medium, lactose is absent. When the lactose is absent, the gene expression is stopped. Because if there is no lactose, then there is no need of the enzymes to break down it. Hence, it is off. It is completely blocked because the repressure which is produced by the regulator gene is on the operator. It is on the operator and it completely blocks the attachment of the RNA polymerase. So it is a tetramer and it blocks the path of the RNA polymerase, and the RNA polymerase cannot attach itself to the promoter. The path is not clear due to the presence of a tetramer, that is a repressor. It is a tetramer, it completely blocks, and the gene expression is stopped. No gene expression taking place because the lactose is not there. When the lactose is not there, now this regulator gene in lac operon is known as lac-I. It is an inhibitor which inhibits the gene expression. So the repressure is over the operator and it completely blocks the gene expression. This is RNA polymerase, and RNA polymerase cannot attach itself to the promoter and to start gene expression. So no synthesis takes place when the lactose is absent in the medium. No synthesis takes place. No synthesis of enzymes and no gene expression. That means there is no gene expression and this is completely shuts off. But when the lactose is present in the medium, when the lactose is present in the medium, we know that when the lactose is not there, the repressor is here. And the RNA polymerase is not binding itself to the promoter, and this regulator gene is continuously producing repressor. It is producing the repressor because one repressor molecule cannot remain there for a longer time. It gets dissociated and a new one is then block this. But when the lactose is present in the medium, the beta-galactosidase lactose is present. It is a disaccharide. It is converted into allolactose by a beta-galactosidase galactosidase present in the medium. This is an isomeric form. In lactose, the bonding between the glucose and galactose is between C1 and C6 of the molecule, sorry, C4 of the molecule, but in allolactose it is C1 and C6. So when the lactose is there in the medium, it is converted into allolactose, and this allolactose attaching itself to the repressor. This is repressor and this is allolactose. The allolactose changes the conformation, that is the shape of the repressor molecule, and due to that the repressor molecule cannot remain there, but it gets detached from the operator. It gets detached from the operator. It has a changed shape, and the whole complex, that is the repressor allolactose complex, gets off from the operator, and now the path is free for the RNA polymerase to get attached to the promoter to carry on, gets attached to the promoter, and now the path is free. The path is free for the RNA polymerase to carry on gene expression, and the three parts of the structural genes, now they start producing the enzymes, the beta-galactosidase, the permease, and transacylase. These are formed because the messenger RNAs are produced, because this operon, it works at the transcriptional level. It is at the level of the transcription. During transcription, this has been modified. So the enzymes are produced and the gene expression is on. So it is with the help of the allolactose that is behaving as an inducer, having the bond between the glucose and the galactose is C1 and C6, and it is then stopping the repressor to remain there. It gets off and clears the path for the RNA polymerase to carry on gene expression. So this is all about the lactose operon. In the next video, I'll describe when the glucose and lactose both are present in the medium, then how the lac operon is operated, is regulated. Thank you.